Hello friends, this video is about acetyl-CoA in fed state as well as in fasting state. I'll be taking, actually many questions are asked about acetyl-CoA, so I am making this separate video for acetyl-CoA. So let's get started. First let's take fed state. In fed state I told you insulin hormone is the main hormone. Insulin activates glycolysis also, insulin activates link reaction also. When insulin will activate glycolysis and link reaction, then lots of acetyl-CoA will be formed. That is link reaction, pyruvate forms acetyl-CoA. The enzyme is pyruvate dehydrogenase complex, PDH complex. This enzyme is activated by insulin. So in fed state, there's conversion of acetyl-CoA. In fed state, this acetyl-CoA can go either into TCA cycle or it can form fats in the body. It will be used for fat synthesis like triglyceride synthesis, fatty acid synthesis and cholesterol synthesis, right? But on the other hand, what happens in fasting state? In fasting or starvation, adipose tissue is the main fuel. So adipose tissue will be broken down to release fatty acids. These fatty acids will go into blood. From blood, they'll be going into liver. In liver, there will be breakdown of this fatty acid known as beta oxidation of fatty acid, which will release acetyl-CoA. Acetyl-CoA can either go into TCA or it can go into ketone body synthesis or it can go into gluconeogenesis. So the fate of acetyl-CoA in fed state is either TCA or fatty acid synthesis or cholesterol synthesis. But the fate of acetyl-CoA in fasting situation is either TCA or ketone body synthesis or activation of gluconeogenesis. Note here, when acetyl-CoA to ketone body arrow is made, it means that acetyl-CoA is converted to ketone bodies. But when acetyl-CoA to gluconeogenesis arrow is made, acetyl-CoA activates the very first step of this pathway of gluconeogenesis. It is not converted to glucose. It is activating this pathway, gluconeogenesis, right? So in fasting state, there is lots of acetyl-CoA. There is lots of acetyl-CoA from beta oxidation of fatty acid. So what will happen to link reaction in fasting state? Link reaction is pyruvate to acetyl-CoA conversion with the help of enzyme pyruvate dehydrogenase complex. Now there is lots of acetyl-CoA here in this situation. The product inhibition will occur here. That is acetyl-CoA, the product of this reaction itself inhibit this reaction so that now pyruvate will accumulate. Now this excess pyruvate will be used for the formation of oxaloacetate. See how many carbon is pyruvate? Pyruvate is 3 carbon compound. Oxaloacetate is 4 carbon compound. From 3 to 4 carbon, we have to add carbon dioxide. So name of enzyme here is pyruvate carboxylase. Pyruvate carboxylase. Any carboxylase use biotin, vitamin B7, ATP and carbon dioxide. And this pyruvate carboxylase reaction is the first step of gluconeogenesis. That is, pyruvate is going to be converted to glucose now. First enzyme is pyruvate carboxylase. And this enzyme is activated by this acetyl-CoA. This is what I have told you in this previous chart, that acetyl-CoA activates the very first step of gluconeogenesis. That is, it activates pyruvate carboxylase enzyme of gluconeogenesis. So summarizing about acetyl-CoA, acetyl-CoA is never glucogenic. It can never form glucose. It is a starting material for ketone body synthesis. Acetyl-CoA is not the first substrate of TCA cycle. Oxaloacetate is the first substrate of TCA cycle. Acetyl-CoA is not the intermediate of TCA cycle because every time in TCA cycle, new acetyl-CoA molecule is used and acetyl-CoA in TCA cycle is getting broken down to 2 carbon dioxide. So this is not the intermediate of TCA cycle. And also, acetyl-CoA is not the carrier of TCA cycle. Again, oxaloacetate is the carrier of TCA cycle. This is it with this video. Best wishes to you. Thanks for watching.